Good morning, everyone. This is the report of Group 3 regarding Experiment 4 Separation of Amino Acids by Paper Chromatography. This presentation consists of an introduction, the methods we use, our results and discussions, and finally our conclusions and recommendations. Paper chromatography is an inexpensive but powerful analytical technique used by scientists to separate and analyze dissolved chemical substances. The first component you need for a chromatography setup is the stationary phase. Typically, this is a porous solid that stays fixed. For this experiment, filter paper was used. Next, we have the mobile phase, a solvent that moves and separates analytes through the stationary phase. We use a mixture of butanol, acetic acid, and distilled water for our setup. For the analytes, we have the amino acids, namely lysine, leucine, tyrosine, glycine, and an unknown. These will travel along the filter paper, being driven by the mobile phase. Once the solvents reach the top of the paper, a spraying reagent will be used to reveal or color the analytes on the solvent front. Ninhydrin is one of the most widely used spraying reagent for amino acids. Once the position of an amino acid is visible on the solvent front, we can now compute for its retention factor. To get this, we first measure the distance traveled by the solvent and the specific solute from the baseline. We divide the distance traveled by the solute over the solvent, and there we have our RF value for that solute. Once we have the different RF values, we can now identify an unknown compound by comparing its RF value with those of the known ones. In 1952, Bush used paper chromatography using alumina paper to identify the different steroids found in mammalian blood and tissues. His data helped fellow scientists understand how steroid secretion is affected by the presence of hormones and neurotransmitters. For this experiment, however, the goal is just to compute for the RF values of the amino acids and identify an unknown acid using the different values we computed.
Okay, so next, for the methods, we divided it into 3 parts for an easier and clearer presentation. The first part is the preparation of the solvent and the chromatography paper. In the lab, a stick-o jar was used as the container of the setup. 24 ml of butanol, 6 ml of glacial acetic acid, and 10 ml of distilled water were mixed carefully, avoiding getting the sides of the jar wet. Then, the jar was covered. Next, for the chromatography paper, a 19 by 14 cm sheet of filter paper was marked 1 cm from the bottom with a light pencil line. Cross marks were put starting from 3.5 cm away from the edges. This was done 5 times. The paper, in the end, had a total of 5 crosses on the line. Shown in this slide is a figure of how the paper looked like after the marks were placed. The dimensions of the filter paper are also indicated. For the next part, we have the application of amino acids. A clean capillary tube was used to apply a spot of amino acid at the center of a cross mark. The spot was dried with a hair dryer before proceeding on to applying more. The amino acid solution was applied 4 times on each cross mark, following this apply, dry, apply routine. It was noted that the diameter of the final spot was only about 1 to 2 millimeters, and that a different capillary tube was used for each amino acid solution. In addition to this, each spot was labeled to identify the amino acid solution applied as well as the unknown applied. Next, the paper was then rolled into a cylinder and stapled, leaving a 1 millimeter overlap at the ends. This rolled up paper was placed in the jar containing the solvent with the crossmark side down. The paper was placed carefully so that it did not touch the sides of the jar. The line with the marks should not get immersed in the solution. The line with the amino acids also should not touch the solvent. The jar was then closed and the rising of the solvent was observed by leaving the setup untouched for 2 hours. The solvent's development was only stopped when the solvent reached a point about 1 centimeter from the top edge of the paper. The last part is the visualization of amino acids. After about 2 hours, the paper was removed from the jar and was left to dry for a while. The point where the solvent reached, the solvent front, was marked with a pencil. The staples of the paper were removed, then the paper was placed on a clean sheet of bond paper. With a spray bottle, the filter paper was coated lightly with the ninhydrin solution. The solution was sprayed across the sheet of filter paper, and after every spray, the paper was dried with a hair dryer. The resulting violet marks were then observed, and the distance the marks reached were measured with a ruler. Next, the RF values for each amino acid were computed using the formula below. The unknown amino acid was identified by then comparing its streaks and RF value with the other amino acids.
So now, let's proceed to the results and discussion of the experiment. After mixing the solvent, which is composed of butanol, acetic acid, and water, its pH was determined using a pH indicator. Here you can see that the solvent has an approximate pH of 3. So here's the result of the separation of amino acids through paper chromatography. The violet spots indicate the positions of the amino acid. The table here shows distance traveled by the amino acid and the solvent along the filter paper and their respective RF values. Note that distance traveled by the solvent is the same for all compounds present. Now let's proceed to the discussion of the results. Before we talk about the factors that affect the speed and distance of migration of the amino acids, let's have a recap about amino acids first. So, amino acids are organic substances that functions primarily as building blocks of protein, as well as regulators of metabolic pathways that are essential for immunity, growth, reproduction, and maintenance. Each amino acid has a basic structure with 5 parts: a central carbon atom, a hydrogen atom, an amino group, and a carboxyl group, and a side chain, or R group, that is made up of varying atoms and is responsible for the uniqueness of every amino acid. The 4 known amino acids used in the experiment are tyrosine, lysine, glycine, and leucine. Next, a typical normal phase chromatography setup consists of a stationary and a mobile phase. The stationary phase for this experiment was a filter paper, while the mobile phase was the mixture of butanol, acetic acid, and water, also known as the BAW solvent. In paper chromatography, the mobile phase is driven up a stationary phase, carrying the solutes along with it. The stationary phase, which is the filter paper or the chromatography paper, are made up of cellulose, which are polymers of the simple sugar glucose. The OH groups in the cellulose enable it to attract water molecules through hydrogen bonding, which is a strong type of dipole-dipole force. Because of this, paper is very polar. And although the solvent is also polar, it is less polar than the filter paper, which contains water droplets in its pores. Next is the mobile phase, which is the BAW solvent, which again is a mixture of butanol, acetic acid, and water. BAW was used since it is a one-phase solvent, or a solvent, which is made up of fewer constituents. These type of solvents are favored because of their stability and ability to provide more accurate results. To visualize the colorless amino acids, the paper was coated with a ninhydrin solution. The amino group reacts with the ninhydrin, producing a type of purple color called Ruhemann's purple. Ninhydrin is often used to visualize fingerprints which have traces of amino acids. After the visualization of amino acids, the distance traveled by each compound and the solvent were measured. The retention values, or the RF values, are then computed using the collected data. RF value is the ratio of the distance traveled by amino acids to the distance traveled by solvent. It indicates the relative extent to which an amino acid moves in the paper chromatography. So RF values are helpful in the identification of an unknown substance, given that a known sample is also applied on the same paper chromatogram. You can identify the composition of an unknown substance by either comparing the streaks left by the amino acids and the unknown compound, or comparing the corresponding RF value or values of the unknown and the possible amino acid or amino acids that it matches with. For our unknown, through visualization, you can see that 2 amino acids separated, and each one has almost the same distance as lysine and tyrosine. They also have close RF values with the said amino acids, so our unknown compound is a mixture of lysine and tyrosine. The difference in RF values of these amino acids can be attributed to their varying structures, which also determine their polarity or affinity to the mobile or stationary phase. Since the mobile phase or the solvent is less polar than the stationary phase or the paper, the amino acid that traveled the greatest distance or has the highest RF value is the least polar, while the amino acid that traveled the shortest distance or has the lowest RF value is the most polar substance. The amino acid that traveled the farthest distance is leucine. Looking at leucine's structure, you can see that it has a purely hydrocarbon side group. Side chains or R groups which have pure hydrocarbon alkyl groups or aromatic, for example benzene ring, are considered to be nonpolar. Amino acids with a higher number of alkyl groups and does not have a polar substituent on the R group is more nonpolar relative to those that do not have the said characteristics. Among all the amino acids, leucine is the only one with a pure hydrocarbon side group. And because of this, it is more attracted to the mobile phase and will travel the greatest distance. On the other hand, the amino acid that traveled the shortest distance is lysine. The structure of lysine shows that it has an amino group in its side chain. Side chains or R groups with functional groups such as alcohol, amines, amides, and acids are more polar compared to those with pure hydrocarbon alkyl chain. Because of this, it is attracted to the more polar stationary phase and will travel the shortest distance. Glycine is also nonpolar, but is less so than leucine since it only has hydrogen on its side chain. On the other hand, tyrosine is a less polar amino acid than lysine because of the presence of a benzyl group. Although tyrosine is more polar than glycine, it traveled a greater distance because the size of its particles are bigger than glycine. Tyrosine has benzene ring on its side chain, while glycine only has hydrogen. So smaller particles may enter the pores of the paper, and those molecules trapped in there are removed from the flow of the mobile phase, hindering further movement, while larger particles like tyrosine, which can be trapped in the pores, can travel with more ease and with greater distance. Another factor that affects the migration of the amino acids are their pKa values and isoelectric points. pKa quantifies acid association through small numbers and is one of the methods used to know the strength of an acid. So the lower the pKa, the stronger the acid. Amino acids can have 2 or 3 pKa values, depending on the structure of their side chain. Amino acids without a functional group on their side chains will have 2 pKa values, one for the carboxyl group and another for the amino group. But if an amino acid has a functional group on its side chain, it will have a third pKa value, an additional one for the said group.
The isoelectric point of an amino acid is the pH level at which it will not migrate in an electric field. To get this, we average the pKa values of an amino acid. For acids with 3 pKa values like lysine, we average the pKa of the carboxyl group in the side chain. The table shows the pKa values of the different components of the amino acids as well as their isoelectric points. Furthermore, lysine is considered a basic amino acid since it has a basic side chain, while the others are considered neutral since they do not have a functional group on their side chains. A similar study was conducted by Franklin and Quashel in 1949. They found out that if the pH of the solvent was close to the isoelectric point of the solutes, then little to no movement will be observed. This implies that if we use a solvent that was slightly less acidic than the butanol acetic acid water mixture, different RF values would have been gathered. In other words, if the solvent we use had a pH of 5.5, which is close to the isoelectric points of the solutes, then the speed at which the solids travel on the paper would be slower, thus resulting in lower RF values.
The last part of our presentation contains our conclusions and recommendations for the experiment. Here, a summary of our key findings as well as suggestions for future iterations of the experiment are presented. An amino acid structure is one of the main explanations for the way they behave in a solvent. In this experiment, we have found that among the 4 amino acids, leucine traveled the farthest and lysine traveled the least. The unknown amino acid, was found to be a mix of tyrosine and lysine. The distance traveled by each of the amino acids is influenced by its structure, which also determines each amino acid's polarity relative to the solvent. The affinity the samples of amino acids have to the mobile phase drive their traveling on the paper. Moreover, the acidity of the mobile phase was also found to possibly have an effect to the distance the analytes traveled. Mobile phase solvent pH levels generally closer or nearer to the determined isoelectric points, or PI, of the analytes allow for slower movement of the analytes leading to lower RF values. In the present experiment, the speed of the amino acids could have been affected by the acidic butanol acetic acid water solvent with its pH of 3, not very near the isoelectric points of the amino acids, which were mostly around 5 to 6, making them travel farther than if the pH of the solvent was around the same range of the values as the amino acids' isoelectric points. Therefore, in general, the distance an analyte travels on a chromatography paper depends primarily on the analyte's polarity in relation to the mobile phase, as well as on the acidity of the solvent used. Overall, a polar mobile phase attracts polar analytes, making them travel farthest, while the nonpolar analytes travel the least. The same principle goes for the nonpolar counterparts as well. Additionally, a closer solvent pH to the analyte's isoelectric points results in the analytes traveling more slowly and having lower RF values. In future practices of this experiment, changing the chromatography technique is recommended in order to get more appropriate and accurate results. Instead of performing paper chromatography, high-performance liquid chromatography could be done. In a study by Neda et al. In 2012, HPLC has been found to be the premier separation method that can be used for amino acid analysis, AAA, from natural products, allowing for the separation and detection by UV absorbance and fluorescence. In HPLC, the mobile phase passes through columns under 10 to 400 atmospheric pressure with a high flow rate. In addition, because the particles used are small, and because high pressure is applied on the rate of solvent flow, the separation power is increased, therefore making the analysis of amino acids finished in a shorter period of time. This is according to a 2016 study done by Koskun. Koskun also explicitly mentioned that HPLC yields perfect results in the separation and identification of amino acids, carbohydrates, lipids, nucleic acids, proteins, steroids, and other biologically active molecules. With this, future iterations of the experiment can use HPLC for faster and more accurate results. Aside from this, it is also recommended that other solvents with varying polarities be used in order to see the different effects of the different solvent polarities to the movement of the analytes. Solvents of different acidities may also be used to more clearly see the effect of the pH on the speed of the analytes along the chromatography paper. Here are references. Thank you for listening.